In brief details on November corona. The novel coronavirus caused severe respiratory illness in 13 adults from Jordan, Qatar, Saudi Arabia, and the United Kingdom. Patients showed symptoms similar to SARS virus, or Severe Acute Respiratory Syndrome, which spread like wildfire in 2002 and 2003, infecting 8,100 people and killing nearly 800. Accurate gene sequencing showed that this new virus isn't SARS or even a known coronavirus that causes colds. Rather, it's a totally new virus that needs to be handled with caution until more is known about it. The new virus is most closely related to coronaviruses that bats carry, but it probably didn't jump directly from bats to people. None of the people who got the disease had direct contact with bats, and the virus is not exactly the same as any known to infect bats. Phylogenetic analysis placed the new SARS-like virus, MERS-CoV, EMC 2012, within the beta-coronavirus genus, where its closest fully sequenced relatives are viruses called BT-CoV HKU4 and BT-CoV HKU5, both of which were originally isolated in Asia from lesser bamboo bats, Tylonicterus pachypus, and Japanese house bats, Pipistrellus abramus, respectively. MERS-CoV EMC 2012 bears only 77% sequence similarity with the BT-CoV HKU5 virus. New coronavirus may have started in bats. But how did it hop to humans? A new study provides more clues to the virus origins, and points to bats as the most likely hosts. In the study, published in the journal The Lancet, the researchers analyzed 10 genome sequences of the novel coronavirus, dubbed 2019 MERS-CoV, obtained from 9 patients in China who were sick with the virus. They found that all 10 of the genome sequences were remarkably similar. They shared more than 99.98% of the same genetic sequence, the authors said. This suggests the virus made its jump to humans very recently, because if that jump had happened long ago, the virus sequences would have differed more, given the fast rate at which viruses tend to mutate and evolve. To learn more about the virus origins, the researchers compared the 2019 MERS-CoV genetic sequence with those in a library of viral sequences. They found that the most closely related viruses were 2 coronaviruses that originated in bats, and both of those coronaviruses shared 88% of their genetic sequence with that of 2019 MERS-CoV. When compared with 2 other coronaviruses known to infect people, SARS and MERS, 2019 MERS-CoV shared about 79% of its genetic sequence with SARS and 50% with MERS. Based on these results, the authors said the 2019 MERS-CoV likely originated in bats. However, no bats was sold at the Huanan seafood market, which suggests that another yet-to-be-identified animal acted as a stepping stone of sorts to transmit the virus to humans. It seems likely that another animal host is acting as an intermediate host between bats and humans, said study co-lead author Guizhe Wu, of the Chinese Center for Disease Control and Prevention. Overall, the outbreak of 2019 MERS-CoV again highlights the hidden virus reservoir in wild animals and their potential to occasionally spill over into human populations, the authors wrote. Coronaviruses are round and surrounded by a halo of spiky proteins, giving them resemblance to a crown or the sun's wispy corona. 4 major categories, or genera, of coronavirus, exist. They are known by the Greek letters alpha, beta, delta, gamma. Only alpha and beta coronaviruses are known to infect people. These viruses spread through the air, and just 4 types, known as 229E, NL63, OC43 and HKU1, are responsible for about 10 to 30% of colds around the world. A new coronavirus spreading in China is similar to SARS, but many of its proteins have important differences. Here, the new virus's similarities, gray, and differences, red, with SARS of the amino acids in a protein that studs the surface of the viruses are shown in 2 views. Those differences may affect the virus's infectivity and how well drugs developed to combat other coronaviruses work against the new one. What makes a virus a coronavirus is only loosely enshrined in its DNA. The coronavirus designation is less about the genetics and more about the way it appears under a microscope, says Brent C. Satterfield, co-founder and chief scientific officer of Code Diagnostics, a company based in Salt Lake City and Gujarat, India, that is developing molecular tests for diagnosing coronavirus infections. Coronavirus's genetic makeup is composed of RNA, a single-stranded chemical cousin of DNA. Viruses in the family often aren't very similar on the genetic level, with some types having more differences between them than humans have from elephants. Research says the new virus's proteins are between 70 and 99% identical to their counterparts in the SARS virus. The naming. The name MERS-CoV 2019, picked by the team of Chinese scientists who isolated and identified the virus, is short for Novel Coronavirus of 2019. It reflects the fact that the virus was first recognized to have infected humans late last year, in seafood and live animal market in Wuhan, and that it belongs to the coronavirus family, a notorious group. Despite the new virus's name, though, and as the people who christened it well know, MERS-CoV 2019 isn't as novel as you might think. Something very much like it was found several years ago in a cave in Yunnan, a province roughly 1,000 miles southwest of Wuhan, by a team of perspicacious researchers, who noted its existence with concern. The inventor. One such scientist is Zhengli Shi, of the Wuhan Institute of Virology, a senior author of the draft paper, not yet peer-reviewed and so far available only in preprint, that gave MERS-CoV 2019 its identity and name. It was Ms. Shi and her collaborators who, back in 2005, showed that the SARS pathogen was a bat virus that had spilled over into people. In a 2017 paper, they set out how, after nearly 5 years of collecting fecal samples from bats in the Yunnan cave, they had found coronaviruses in multiple individuals of 4 different species of bats, including one called the intermediate horseshoe bat, because of the half-oval flap of skin protruding like a saucer around its nostrils. The genome of that virus, Ms. Shi and her colleagues have now announced, is 96% identical to the Wuhan virus that has recently been found in humans. And those two constitute a pair distinct from all other known coronaviruses, including the one that causes SARS. In this sense, MERS-CoV 2019 is novel, and possibly even more dangerous to humans than the other coronaviruses. Current circumstances. Current circumstances include a perilous trade in wildlife for food, with supply chains stretching through Asia, Africa and to a lesser extent, and elsewhere. That trade has now been outlawed in China, on a temporary basis, but it was also outlawed during SARS, then allowed to resume, with bats, civets, porcupines, turtles, bamboo rats, many kinds of birds and other animals piled together in markets such as the one in Wuhan. Current circumstances also include 7.6 billion hungry humans, some of the impoverished and desperate for protein, some affluent and wasteful and empowered to travel every which way by aeroplane. These factors are unprecedented on planet Earth. We know from the fossil record, by the absence of evidence, that no large-bodied animal has ever been nearly as abundant as humans are now, let alone so effective at aggregating resources. And one consequence of that abundance, that power, and the consequent ecological disturbances is increasing viral exchanges, first from animal to human, then from human to human, sometimes on a pandemic scale. We are responsible. We invade tropical forests and other wild landscapes, which harbor so many species of animals and plants, and within those creatures, so many unknown viruses. We cut the trees, we kill the animals or cage them and send them to markets. We disrupt ecosystems, and we shake viruses loose from their natural hosts. When that happens, they need a new host. Often, we're it. The list of such viruses emerging into humans sounds like a grim drumbeat. Marchupo, Bolivia, 1961, Marburg, Germany, 1967, Ebola, Zaire, and Sudan, 1976, HIV, recognized in New York and California, 1981, a form of Hunta, now known as Sin Nombre, southwestern United States, 1993, Hendra, Australia, 1994, bird flu, Hong Kong, 1997, Nipah, Malaysia, 1998, West Nile, New York, 1999, SARS, China, 2002-3, MERS, Saudi Arabia, 2012, Ebola again, West Africa, 2014. And that's just a selection. Now we have nCoV 2019, the latest thump on the drum. How do new viruses emerge? Human health, animal health and the state of ecosystems are inextricably linked. 70 to 80% of emerging and re-emerging infectious diseases are known to be of zoonotic origin, meaning they can be transmitted between animals and humans. Population growth, climate change, increasing urbanization, and international travel and migration all increase the risk for emergence and spread of respiratory pathogens. The hope. Fortunately, current circumstances also include brilliant, dedicated scientists and outbreak response medical people, such as many at the Wuhan Institute of Virology, EcoHealth Alliance, the Chinese CDC and numerous other institutions. These are the people who go into bat caves, swamps and high-security containment laboratories, often risking their lives, to bring out bat feces and blood and other precious evidence to study genomic sequences and answer the key questions. We are faced with two mortal challenges, in the short term and the long term. Short term, we must do everything we can, with intelligence, calm and a full commitment of resources, to contain and extinguish the nCoV 2019 outbreak before it becomes, as it could, a devastating global pandemic. Long term, we must remember when the dust settles, that nCoV 2019 was not a novel event or a misfortune that befell us. It is, part of a pattern of choices that we humans are making. Risk assessment. Outbreaks of novel virus infections among people are always of public health concern. The risk from these outbreaks depends on characteristics of the virus, including whether and how well it spreads between people, the severity of resulting illness, and the medical or other measures available to control the impact of the virus, for example, vaccine or treatment medications. This is a serious public health threat. The fact that this virus has caused severe illness and sustained person-to-person spread in China is concerning, but it's unclear how the situation in the United Kingdom will unfold at this time. The risk to individuals is dependent on exposure. At this time, some people will have an increased risk of infection, for example, healthcare workers caring for 2019 nCoV patients and other close contacts. The timeline of 2019 nCoV. The 31st of December 2019, China alerts the World Health Organization, WHO, about a spate of pneumonia-like cases in Wuhan. The 1st of January 2020, the seafood/animal market believed to be at the center of the outbreak is closed. 9 January, the WHO says the infection is caused by a new type of coronavirus. 10 January, China shares the genetic code of the new virus. 11 January, scientists start working on a vaccine, and the first death confirmed. 13 January, virus spreads abroad for the first time, with a case in Thailand. 17 January, Thailand confirms the second imported case of 2019 nCoV in the country, bringing the total exported cases from China to 3. 20 January, South Korea confirms its first case of 2019 nCoV. The patient did not report visiting markets in Wuhan. 21 January, WHO confirms human-to-human transmission of the virus. The total number of cases is now 222, including infections among healthcare workers. Chinese authorities have also reported the fourth death. 25 January, the 2019 MERS-CoV reaches more countries. Australia confirms the first case of 2019 MERS-CoV in the continent, with health authorities announcing 3 more cases later in the day. France confirms 3 cases, the first in Europe. Malaysia also reports the first 4 cases. Canada reports its first case. 1 February, in China, confirmed cases now total 14,380, and the death toll rises above 300. 2 February, the first 2019 MERS-CoV death outside China is reported in the Philippines. 6 February, Japan announces a $10 million contribution for WHO's 2019 MERS-CoV outbreak response fund to support countries with weak health systems as they prepare for the potential spread of the virus. 11 February, WHO assigns the novel coronavirus its official name, COVID-19. 15 February, France reports the first death from COVID-19 outside of Asia, an 80-year-old tourist from Hubei province. 19 February, the death toll from COVID-19 surpasses 2,000. 25 February, Algeria reports its first case of COVID-19, an Italian adult, who arrived in the country on February 17. This is the second confirmed case on the African continent. 27 February, the number of infections globally continues to grow. There are 3,474 cases of COVID-19 including 54 deaths, outside of China in 44 countries. 29 February, the World Health Organization updates its guidance on travel restrictions. 1 March, the United Nations releases $15 million from its Central Emergency Response Fund to the World Health Organization and UNICEF to support vulnerable countries in areas including monitoring the spread of COVID-19, investigating cases, and operating national laboratories. 2 March, the United States commits $37 million from the Emergency Reserve Fund for Contagious Infectious Diseases at the U.S. Agency for International Development for countries affected by COVID-19 or at high risk of its spread. 3 March, the World Bank Group commits $12 billion in immediate support to help countries strengthen health systems and to help cope with the economic impacts of the COVID-19 outbreak. 4 March, the Global Fund to Fight AIDS, Tuberculosis and Malaria says countries may use savings from their existing grants for COVID-19 response, with a limit of up to 5% of the grant's total value. Figure, a computer artwork of a coronavirus, named after the corona, or crown, of surface proteins, outer dots, that are used to penetrate a host cell. Airlines are already required to report passengers who show signs of infectious disease, including fever, sweats or chills, so sick travelers can be met by biosecurity officers when they arrive in Australia to be assessed. Figure, thermal scanners are sometimes used to identify travelers with fever, but they can be unreliable, Dr. Cheng says. There's an ever-increasing diversity of animal coronavirus species, especially in bats. So the likelihood of viral genetic recombination leading to future outbreaks is high. The threat of future pandemics is real as highly pathogenic coronaviruses continue to spill over from animal sources into the human population. Row, reproductive number. Misdiagnosis of future outbreaks poses an additional threat to healthcare workers, with hospital-based spread to other patients putting further pressure on already strained healthcare systems. It depends on the coronavirus, but neither SARS nor MERS have been able to sustain human-to-human transmission the way influenza viruses can. That's because the viruses haven't fully adapted to infect humans. Epidemiologists are frantically calculating how infectious the new virus is, says an experienced scientist. The attack rate or transmissibility, how rapidly the disease spreads, of a virus is indicated by its reproductive number, rho, pronounced R0 or R0, which represents the average number of people who will catch the disease from a single infected person. SARS, for instance, had an R0 between 2 and 5, meaning that in a fully susceptible population an infected person could potentially spread the virus to 2 to 5 others. Highly contagious measles, in comparison, has a R0 from 12 to 18. Estimates for the infectivity of the new virus range from the WHO's estimate of 1.4 to 2.5 to a much bigger 3.6 to 4.0 calculation from Jonathan Reed of Lancaster University in England and colleagues. Reed's group estimates that only about 5.1% of cases in Wuhan have been identified. Many people may have had only mild symptoms or none at all. Those people probably wouldn't go to the doctor and get tested for the virus. A pathogen's harmfulness is determined by the combination of its transmission rate, rho, reproductive number, and its case fatality rate, CFR, so we'll examine these two parameters below. For the Wuhan coronavirus, this parameter is estimated to be between 2.0 and 3.1, according to preliminary studies, with one study providing an estimate of 2.6 with an uncertainty range of 1.5 to 3.5. The WHO noted on January 23 that human-to-human transmission was occurring and a preliminary rho estimate of 1.4 to 2.5. Based on these numbers, on average, every case of Wuhan coronavirus would create between 2 and 3 new cases. An outbreak with a reproductive number of below 1 will gradually disappear. For comparison, the rho for the common flu is 1.3, and for SARS it was 2.0. Fatality rate, case fatality ratio or CFR, of the Wuhan coronavirus. Wuhan novel coronavirus, 2019 nCoV, mortality rate. The novel coronavirus case fatality rate, CFR, is currently estimated at around 2%, the World Health Organization, WHO, said on Wednesday, January 29, 2020. However, the WHO specified that this is a very early and provisional estimate that may change. Surveillance is increasing, within China but also globally, but at the moment, the only number currently is known is how many people have died out of those who have been reported to the WHO. It is therefore very early to make any conclusive statements about what the overall mortality rate will be for the novel coronavirus, according to the World Health Organization. A prior estimate had put that number at 3%, between 2% and 4%. Fatality rate can change as a virus can mutate, according to epidemiologists. Once the province Hubei, where the epicenter Wuhan is situated, is removed from the calculation, the national, China, mortality rate drops to 0.3%. Within the Hubei province, the mortality rate is about 1% when excluding the city of Wuhan, where it is 5.5%. Comparison with other viruses. For comparison, the case fatality rate with seasonal flu is less than 0.01%, 1 death per every 10,000 cases. The mortality rate for SARS was 10% and for MERS 34%. Age and conditions of coronavirus cases. According to the WHO Situation Report No. 7 issued on January 27, the median age of cases detected outside of China is 45 years, ranging from 2 to 74 years. 71% of the cases were male. The WHO, in its Mythbusters FAQs, addresses the question, does the new coronavirus affect older people, or are younger people also susceptible? By answering that, people of all ages can be infected by the new coronavirus, 2019 nCoV. Older people and people with pre-existing medical conditions, such as asthma, diabetes, heart disease, appear to be more vulnerable to becoming severely ill with the virus. WHO Risk Assessment, Global Emergency. On January 30, the World Health Organization has declared the coronavirus outbreak a global public health emergency.